Hi, my name is Ellen Fu, and I want to give you guys a very quick and brief introduction to stem cells. As most people know, stem cells are blank slates within an organism that can produce many different types of cells. However, there are actually categorizations of those stem cells depending on how committed to their cell lineage that they are. First, there are totipotent stem cells that can produce any other type of cell within the organism and can produce more totipotent stem cells. Only embryonic stem cells extracted from blastocysts are totipotent. Second, there are pluripotent stem cells, which can produce any other type of cell except for totipotent stem cells. Lastly, multipotent stem cells are relatively specialized. While they can still produce many different types of cells, they can only do so within a specific tissue type. Differentiation is the process where stem cells start to express certain genes and not others so they can start to perform more specific functions. These factors include chemical factors, the lineage of the stem cell, and the microenvironment. Chemical factors include hormones and other growth factor chemicals, and cell lineage means what tissue type the multipotent stem cells come from. And lastly, microenvironment can include temperature, the surface type, and even the texture of the surface that the stem cells are growing on. There can be many useful applications of stem cells if they can be fully understood, and this includes generating new tissues for replacement, like cartilage and organs, and this is especially important to the medical industry because this can shorten the lengthy process of finding a donor, and the scientists can control the genetic expression of the stem cells so the specific and exact type of organ they want can be produced. Second, stem cells could be used to study diseases and cures. There is a process called gene therapy where a working gene is transplanted into a multipotent stem cell that comes from a specific malfunctioning organ from the patient. Then, these multipotent stem cells are transplanted into the patient so they can produce more working cells. As good as everything might sound, there are many obstacles in stem cell research. From the ethical side, human embryonic stem cells are the most useful in research. However, by extracting them from embryos and doing research on them, some people think that it's essentially taking apart what could have become a baby. And second, the mass culturing of stem cells and then throwing them away when they are no longer useful seems ethically cruel to people because it has a sense of playing God by creating then throwing away life. From the technical side, stem cells are similar to cancer cells in the way that they divide without hindrance. Scientists have actually tried to insert cancer genes into normal cells to revert them back to the stem cell status. But these cells cannot be applied in actual medical practices yet because of all the unknown effects. Thank you for listening. I hope you learned more or refreshed your basic knowledge of stem cells through my video.